Good evening, ma'am.
Good evening, sir.
Good evening.
Sir, you can start now.
Yes, ma'am. Yes. Thank you. So, good evening, my dear students.
Good evening, sir.
Good evening, Dhanalakshmi. Okay. Last class, what we have discussed. You know very about.
Sir.
Good evening, Ajay. Good evening. Okay. So, last class, what we have discussed already, you know very about what is by DNA and what are the content of the DNA and historical aspect, who discovered DNA and gave a name nuclein, and who discovered a double standard helical structure, and on the basis of what, all those things already we discussed, isn't it? See here, so the DNA molecule consists of three component, otherwise called as a nucleotide. Okay, so that things already we discussed. One is sugar component, another one is called as nitrogen bases, and third one is called as phosphate. These three components present in a polypeptide chain is called as what is the nucleotide. Anyone is, particularly the phosphoric acid is absence of phosphoric acid, only the nitrogen base as well as a sugar molecule present in it, it is called as nucleoside. Okay, already we discussed. And purine and pyrimidine, what are the structure is there? Already we studied. So what about that? Their chemical composition. What will be the chemical name of the particular molecules? Adenine, 6-aminopurine, guanine, 2-amino-6-oxypurine, cytosine, 4-amino, 2-oxypyrimidine, and thiamine, 2,4-dioxy, and 5-methylpyrimidine. Uracil also left here. Already I told you it's for your homework. Okay, please. Okay. See that these are the structures. These are the structure. These are all are called as what is that? And 6 carbon atom. Okay, these are all pyrimidine, cytosine, thiamine. So cytosine, second letter is Y. Thiamine is also a second letter, or third letter is Y. Okay, so these are all identification of the pyrimidine and purine. Difference between these two molecules, these two nitrogen bases. Okay, my dear. Okay. Go to the next one. This is the things. Normally, the DNA containing the purine molecule is, what is that, you know? Adenine and guanines. It's a purine molecule. Heterocyclic nature, 9 carbon compound, all those things already you know about. But the RNA is also same. See that? Adenine and guanine. But pyrimidine, what happened, you know, the thiamine and cytosine is their adenine molecules always combine with the molecules of thiamine with the help of the bond, hydrogen bond. Adenine combine with the molecules of thiamine with the double bond here. Guanine combine with the cytosine with the triple hydrogen bond. Okay, so here the RNA molecules, ribose molecules, which one is replaced? No, thiamine is not present in it. Instead of thiamine, uracil is present in it. Okay, so now the adenine molecules joined with the double bond, similar to that of the DNA molecules. Adenine combined with the molecules of uracil only, but the RNA molecules, thiamine is not present. The DNA molecule, uracil is not present. Okay, these two are replaced. Note it down, my dear. This is a common one. This is the common nitrogen bases. Particularly, the uracil is replaced by thiamine. Okay, so adenine combined with the molecules of uracil with double bond. Guanine combined with the molecules of cytosine, triple bond. When the translation process, what happened, you know, how many amino acids are present in it, in your body? Any one of you, my dears? How many amino acids?
Sir, 20, sir.
Pardon, pa?
20, sir.
20, exactly. Very good. Exactly, correct. But the 20 amino acids exist in how many codon? How many codon? Do you know? 3 combinations, 3 nitrogen bases united together form as a single amino acid. It encoded a single amino acid. When you are going to study the translation process, we will calculate it. Okay. So AUG is the starting codon. AUG is the starting codon. We'll discuss that things also in future. Okay. During the translation process. Are you clear in this part, my dear? Okay. So where there is a change, just note it down, my dear. Adenine is also common, both, but the thymine and uracil is also replaced. Adenine, thymine. Here, adenine and uracil. But guanine and cytosine, there is no change in both cases. Okay. So this is a common one. This is also asked in the NEET question paper, and I think 2017. Just to go and check it. 17 or 18 already. I have sent the 5 to 6 year question paper, original question paper in your link. I mean WhatsApp. Just go and check it and practice it every day. Each and daily you have to take the question paper and then read and then answer it. Please. Okay. So this is the important things. See that one. So already we know purine. These are the two things is the heterocyclic nature. This is also a heterocyclic. They told heterocyclic like that. But the pyrimidine is a 6-carbon atom. Aromatic. Okay. That thing's also already discussed. Phosphate group. Excuse me. Phosphate group. The inorganic salt of phosphorus forms backbone of the polynucleotide chain along with the sugar molecules. Phosphate molecules, nitrogen base, as well as the sugar molecule. These are all called as what is a nucleotide. It is repeated more than 1,100. That's why the DNA is also called as polynucleotide chain. DNA is also called as what is that? Polynucleotide chain. Okay. So see the next slide, my dear. So this is the only one. What is that? Combination. What are the combination present here? So this is called as nitrogen base. Here there is a adenine. Okay. This is 5-carbon sugar molecules. The nitrogen base came here. Okay. And also the sugar molecules also comes here. And what about the phosphate molecules also comes here? So the 3 component is also present. The joint will be like this. Base with the sugar molecule and phosphate molecule. This is the one strand. The opposite strand is also there. He's here, the 5' end. 5'. Okay. 5' join with the phosphate molecule with the help of which one? Glucose molecules join with the phosphate molecule. This joint is called as what is known glycosidic, phosphoglycosidic linkage. Sugar molecules, 5th carbon atom united with the phosphate molecules. Don't forget, my dear. So this bond is called as what is known glycosidic linkage. See that one. Next one we can easily identify. Formation of the polynucleotide takes place using the following linkages. Without these bonds, take an example: N-glycosidic bond, phosphodiacid bond, and 3'5' phosphodiacid bond. These are the 3 bond is essential for the construction of the double-stranded molecule DNA. What are the bond is very, very important, N-glycosidic bond. Already we discussed. Glycosidic. Glycosidic means glucose molecules linked with the, which one, amino group. Phosphodiester bond and also phosphate group also linked with the nucleotide. Multiple nucleotide are joined together through the 3' to 5' Already what, but the DNA molecule is consist of 2 strand is there, isn't it? DNA molecule consists of 2 strand is there. Okay, just see the next slide. It will be better, I think. See that. So this is the sugar molecule. This is the base, nitrogen base. This is the one strand. Okay, starting from the one strand. The same structure present the opposite side also. Okay, so the nitrogen base adenine. This is considered as a guanine. Again guanine, like that. The base is connected with the sugar molecules. The base is connected with the sugar molecule and also the phosphate molecules. These 3 is called as what is that? Nucleotide. The 3 is called as nucleotide. Likewise, the opposite side also there is a nitrogen base is there, and also sugar molecule and phosphate is also there. Okay, because it's only one strand here mentioned. The opposite is also there. A complementary strand is also there. This is started from 5' to 3', opposite side 3' to 5'. See that one. It is better for your identification. See. See, so this is a DNA structure. So this is the DNA structure like this. Imagine. This is started from the 5 dash to 3 dash. Here, so 3 dash and 5 dash. This structure, okay. So in between there is a nitrogen bases here. Here only. The nitrogen bases, what are they? Nitrogen bases already you study, no? It is a DNA molecule, means you know about adenine molecule combined with the thymine. Guanine molecules combined with the triple bond hydrogen bond. It is called as, what is that? Cytosine. Here the joining is, what is that, you know? The sugar molecules, already you know about the sugar molecules also I give different colors. Then only you can easily identify it. So this is the pentose sugar molecules. Pentose sugar molecule. So nitrogen base comes here. Only one nitrogen is also. This is a stand, isn't it? This is consists of 3 dash to 5 dash stand. It is a polynucleotide chain. So here the nitrogen base comes here, followed by phosphate, followed by the sugar molecules, and addition with this, which one you know? See that one? This is the phosphate. So in this part, there is a phosphate molecule is there. Okay. So these portion only up to this level. Okay, up to this portion is called as, what is that, you know? Nucleotide, the phosphate, sugar molecule, and nitrogen bases. Likewise, other strands also having, what is that? The same structure. Okay, there is a phosphate molecules connected with the, okay, so the sugar molecules here. So the sugar molecules, you have to put the sugar molecule structure here. Instead of sugar molecule, I write here sugar. Connect with the nitrogen bases. So in between these two nitrogen bases, there is a bond. Okay, when you are heating this, what is that? When you are heating this DNA molecules, when you are giving the heat, what happen? No, the adenine combination, you know, adenine combined with the thymine and guanine combined with the cytosine. This bond is called as nitrogen bond. Okay, here double bond is there, here triple bond is there. Suppose when you are giving the heat like this, you know, this bond is broke. This bond is break. As a result, how it will be, you know, the single strand separately you will get it. The single strand is, we will get it separate manner. Because the bond is broken here. See here, this bond is broken means the adenine as well as the sugar molecule and phosphate, it will be separated like this. Okay, here the see here, see that. See that. So this is the adenine. Okay. So here the already you know, but there is a sugar molecule is there, and followed by the phosphate. This is the one strand, this part. Okay. And then opposite side, what is that? Adenine opposite, which one is there? Present, you know, thymine, as well as the sugar molecules, as well as the phosphate. These two strands are completely separated without this bond formation. When you are giving the heat, okay, the condition is called as, what is that? Heat up to the temperature up to the 60 degrees Celsius or 50 degrees Celsius. What happened? You know, the DNA structure will be denatured and split into two. What is that? Polypeptide chain separately. Okay. Suppose when you are giving a cooling condition, okay, heat condition, the hydrogen bonds are weak bond, so it is easily broke. So what happened? You know, automatically it divided into two strand separate manner. Okay. When you are giving the cooling condition means that condition is called as annealing. Note it down, my dear students. Okay. When you are giving the or reduce the temperature, that condition is called as annealing. That time what happened? You know, it's once again rejoined together and form as a what is a helical structure? Form as a double helical structure, joined together again. So when you are giving the heat, what happened in a split? When you are giving the cool conditions, okay, cooling condition, what happened? Automatically it will be rejoin. Cooling conditions, what happened? Rejoin and form as a similar structure like this. This condition is called as, when you are giving the cooling condition, that condition is called as, what is it in annealing? Annealing, I will type here, then only it will be better for you. Here, in this condition, so annealing. Okay, this is the process, annealing. When you are giving the annealing process, otherwise called as temperature reduce or cooling process, what happened? The DNA molecules united together form as a double structure. Okay. Okay. See that one. Why we are studying, we will study, it will be essential. So that's why I told you. Okay. Okay. See here. Okay. See, the formation of the polynucleotide takes place using the following linkage. Nitrogen base is linked to the pentose sugar. Note it down, my dear student. Nitrogen base, that's why N. Nitrogen means amino. Base is linked to the pentose sugar. It is a sugar molecule. That's why it is called as glucose or glycose. Okay. So, through a N-glycosidic bond to form a nucleoside. Nitrogen base, sugar molecule joined together with the help of the bond is called as N-glycosidic bond. So as a result, you will get it nucleoside. Here there is no phosphate, isn't it? The question is asked here. The N-glycoside bond is formation takes place within the following component. Which one? Nitrogen base and phosphate. Nitrogen base and pentose. Pentose and phosphate molecules. Like that, they may ask the question. Don't forget, my dear, N-glycosidic means N denoted as nitrogen base and glycosidic means sugar molecule. These two combinations, these two joints linked with the help of the bond is called as N-glycosidic bond. See that? See that? So nitrogen base as well as a sugar molecule. This bond is there, in between this bond. This bond is called as N-glycosidic bond because the amino group, this one is called as glucose. So N-glycosidic bond. Where there is a bond between the sugar molecule as well as a phosphate, what is that? See that? The phosphate group is linked to the fifth carbon atom or hydroxyl of the nucleoside through phosphoester bond to form a nucleotide. See? Hydrogen base linked with the sugar molecule N-glycosidic. Okay. The fifth carbon atom of the sugar molecule combined with the phosphate OH molecule. This combination is called as ester bond. Phosphate molecule with sugar, ester bond. Phosphate molecules with the sugar, ester bond. Sugar molecule with the nitrogen base is called as N-glycosidic bond. This is the N-glycosidic. This is the phosphate bond. Okay. Phosphodiester. Why is called as diester? Here the one ester bond is in this area. Similar one is also opposite side. So two phosphate molecules also leading to the DNA strand. So that's why diester bond. See that one? The multiple nucleotide are joined together through the 3' 5' phosphodiester bond to form a polynucleotide. Likewise, more than one glucose molecules, more than one nitrogen bases. That is the things linked here. See, this is the one step. Sugar and phosphate. Here, diester bond. Sugar and base, glycosidic bond. Likewise, sugar and phosphate. What is that? Ester bond. Here, sugar and so likewise, there are so many. What is that? So many combination is there. That combination is called as polynucleotide formation. This is the nucleotide formation. Each and every DNA molecule, there are a number of nucleotide molecules present in it. Okay, so this is the formation. Nucleotide means we need three components. What are they? Nitrogen base, sugar, and phosphate. How they link? a base as well as a nitrogen base as well as the sugar molecule, N glycosidic bond, glycosidic, sugar as well as the base. In this part, ester bond, phosphate with the sugar molecule. Likewise, many components, this is number, denoted as number of molecules united together in a polypeptide chain. That is called as polynucleotide. Okay. So this is the structure. See here. So many things. This is the single strand nucleotide. Single strand nucleotide only. See the nitrogen base is the sugar molecules automatically in overlap, but this is the RNA structure. This is the DNA structure. Double standard DNA helical structure. This was discovered by the name of the scientist already in overlap about 1953. What is that? Watson and Crick. So RNA molecule is a single standard molecule, single standard. Okay. Sometimes it may be double standard. Take an examples of corona and take an examples of HIV. It contains a double standard or single standard molecules. Okay. Sometimes it may be double standard. Take an examples of cauliflower mosaic virus. Cauliflower mosaic virus. Cauliflower mosaic virus is one of the plant viruses, but they contain, what is that? Double standard RNA molecule. Sometimes it may be a DNA. Okay, some of the molecules. Take an example. I will tell you one by one. HIV is which molecule present in it, my dear? HIV. HIV. Which nucleic acid present inside the virus?
Sir, the RNA, sir.
Very good. How many copies? 2 copies of RNA with 2 copies of reverse transcriptase enzyme. Okay. Okay. That is a animal virus, isn't it? Plant virus, what happens? Sometimes it may be a single standard, sometimes it may be a double standard. Here you must note what are the difference between DNA and RNA. I mean, DNA double standard structure, RNA single standard. But here, what are the nitrogen bases present in it? Already you know very well. Thymine, cytosine, adenine, guanine. Here, which one you know? The uracil is replaced by thymine. So remaining things are same. RNA. Okay. So this is the lowest molecular weight. Lowest molecular weight. Okay. When we are considering nitrogen bases. Okay. So this is the very, very important thing. See that bond. How it will be united? See, already I told you, this is the hydrogen bond. One nitrogen base is there, and this is the nitrogen base. Connected with the, which one you know? Another nitrogen. So take an example. So see that this is the double structure, isn't it? Triple bond is there. Hydrogen bond. See here, hydrogen bond here, hydrogen bond here. So when there is a triple bond is here, it is considered as here, which one you know? Guanine and cytosine. See that guanine G. See the cytosine connected with the triple hydrogen bond. Okay. See here, adenine combined with the molecules of thymine. There is only 2 hydrogen bond is there. Okay, 2 hydrogen bond is there. The combination. See here, already I told you. This is the combination. So the base is here. Sugar molecule. The phosphate is also. This one structure is called as nucleotide. More than one structure is called as what is that? Polynucleotide. Okay. The phosphodiester bond. See, listen here. Here the one phosphate molecule is there, and another one is also here. In between, there is a 2 phosphate molecules are united. One phosphate molecule united with the one side with the above sugar molecule. Another side there is a below sugar molecule. So the bond is in between the 2 sugar molecules. The only one phosphate is a participate. That's why it considers a phosphodiester bond. The 2 sugar molecule united with the phosphate with the help of the bond is called as phosphodiester bond. Di means 2 sugar molecule. The ester bond is here. Which component united the sugar molecules? You know, phosphate molecules. So that is the reason. See, don't forget, my dear. Guanine, cytosine, 3 hydrogen bond. Adenine, thymine, 2 hydrogen bond. What is by phosphodiester bond means 2 sugar molecules united with the phosphate molecule with the help of the bond is called as phosphodiester bond. Simply. Okay. Okay. See that one. So this is another important thing, nucleotide and nucleic acid nomenclature. Here there are 4 column has given. One is called as base. What are the bases there? Already know. Purine, adenine, guanine. Pyrimidine, cytosine, thymine, uracil. These are the things. Nucleoside means absence of phosphoric acid. What is the meaning of this one? Adenosine. That is also adenosine. Okay. Okay. So nucleotide means adenylated. It's present in the RNA. Adenine, adenosine, adenylated. Okay, RNA. So deoxyribose means DNA. So deoxyadenosine, same one, you have to add the deoxy because it's present in the DNA. Deoxyribonucleic acid. That's why you have to put before the adenosine, you have to put deoxy because it's present in the DNA. So here RNA. Deoxyadenosine. It converted to nucleotide, deoxyadenylate. So DNA. Guanosine, guanylate, RNA. Deoxyguanosine, deoxyguanylate, DNA. This is purine. Pyrimidine means similar to that one. Cytosine, cytidine, cytidylate, RNA. Deoxycytidine, deoxycytidylate, DNA. Thymidine, thymidinylate, DNA. Deoxy means you know what about. Okay, RNA is not here because thymine is absent in the RNA. So RNA it will not produce it, isn't it? Uracil, uridine, uridylate, RNA. This is very, very important. So many times they were asked the question, the uracil conversion of nucleoside, nucleotide. Are you clear in this part, my dear? Dhanalakshmi?
Yes, sir. Understood, sir.
Okay, okay. See, once again, if you want to repeat. Okay, anyhow. So purine, adenine, guanine, pyrimidine, cytosine, thymine, and uracil. Nucleoside, what are the composition? So you have to put sign. Each one is a sign. Cytidine is also different. Okay, uridine is also different. Here you have to add the adenylate, late, L-A-T. We all are having the suffix is called as late, adenylate, thymidylate, deoxycytidylate, okay, uridylate, like that. Okay, where it present. Deoxy means DNA, cytidine means RNA. Okay, likewise. See that? This is another combination. Adenine combined with the molecules of ribose sugar, adenosines. This is nucleoside. Adenine is a nitrogen base, sugar molecule ribose sugar. Ribose means RNA. Okay. The formation is called as adenosine. After the formation of adenosine, the nucleoside, it combined with the phosphate molecules. This is called as nucleoside. Okay. What happened here? RNA, adenine nitrogen base, combined with the molecules of sugar. So with the help of the N-glycosidic bond is here. So I will get adenosine. Adenosine is nothing but nucleoside only. Here there is no phosphate. So nucleoside. So after the formation of nucleoside, the adenosine is also, see, the same things is here. Adenosine, phosphate linked, what happened? No, it formed as a adenylated. So this is called as nucleotides. Here what happened? Tide means 3 components. Adenosine, ribose sugar, phosphate. 3 component is here. So nucleotide. Likewise, DNA also, adenine, deoxyribose sugars. Here, ribose sugar, a deoxyribose sugar. You will get deoxyadenosine. This is a nucleoside. The deoxyadenosine again joined with the or combined with the phosphate molecule, you will get deoxyadenylates. This is nucleotide. Likewise, you have to practice all the component. Adenine only I have mentioned here. You have to practice all those things already. There is a combination here. Okay, my dear. So you practice it. This is also very, very important. Sometimes they may give a question, which one is the following is correctly given. Okay, direct question, they may ask the question. Nucleotide or nucleoside, like that, they may ask the questions. Okay, this is the DNA structure. So this is the strand. Already you know about this is the helical structure, just like a ladder. Okay, ladder twisted like structure, this is the double helical structure. There is a sugar molecule. See that sugar molecules is here. This colored one. In between there is a small line. Different color has given. These are all are nitrogen bases. Okay. And corresponding base pair is also adenine, thymine, guanine, cytosine, like that. Okay. But the bond is in between these two. Here sugar molecule is there, here is also. The bond between these two. Then only the structure will be maintained. Otherwise, it will not maintain the chemical structure. It can be easily broken. Okay. See that next one. You can see that. Okay. Next important things. Don't forget, my dear. Without this part, there is no question in the molecular basis of inheritance. Already know very well about the Chargaff rules. Without Chargaff rules, there is no any DNA molecule. Okay, structures. What they did? How? See here, the E. Chargaff and his colleagues in 1949 analyzed the composition of the DNA from various sources by quantitative chromatographic method. What is the method name is called as quantitative? How much amount? Chromatography, colored one. Okay, used for the separation of the enzymes, amino acids, all those things, protein, lipids, whatever it may be, we can easily identify it. Only for the quantitative separation. Qualitative means we know very about. That is called as, what is that? Electrophoresis. DNA separations, gel electrophoresis. Chromatography is also different types is there. Thin layer chromatography, column chromatography. So many chromatography method is also there. Okay, we will study that things on features. See, the Chargaff and his colleagues in 1949, what they did, they analyzed the composition of DNA from various sources by quantitative chromatographic method. This observation are called as Chargaff rules. Okay, regardless of the source, the purine and pyrimidine base occurs in equal amount in DNA molecules. That is very, very important. What is that? What is he stated? The statement, purine and pyrimidine bases occurs in equal amount in a DNA molecule. For example, take an example, adenine 15 is there, So adenine always combined with the molecules of thymine. So adenine 15 means the thymine is also 15. Guanine 60 means cytosine is also 60. That is the reason. According to this one, the max oriented calculation also sometimes they asked in this part. I will tell you. Okay. So the amount of adenine is equivalent to the amount of thymidine. So A that is equal to T that is equal to 1. Likewise, guanine that is equal to C that is equal to 1. So guanosine, cytosine, adenine, thymine. These are all very, very important. Max oriented questions asked in this part only. See that one? The amount of cytosine is equivalent to the guanine, C by G that is equal to 1. Thus, there is a base equivalent in DNA. Take an example, any one of the DNA molecule, take an example of animals or plants or whatever it may be, the adenine molecules, guanine molecules, cytosine and guanine, and adenine, guanine, adenine and thymine, guanine and cytosine, both are equally present. Okay. Okay, you got it? Okay. Suppose if it is any variations, what happens? See here, the base ratio, adenosine plus thymine, guanosine plus cytosine, however, is rarely equal to 1 and varies with the different species from 0.4 to 1.9. It's mostly found in low in microorganisms and high in higher animals, but the ratio is constant for a species. This is the differences. Suppose the changes occurs in this part, the guanine and cytosine, thymine or whatever it may be. What happened, just like a R.D. Weinberg law, P squared plus Q squared that is equal to 1. So P squared is equal to 1, Q squared is equal to 1. That formula is also applicable here. Okay. So likewise, this component is also similar. Suppose if it is any variation in there, the variation result, it will not provide 1. It will not give 1. Suppose the variation is less 1 or the higher 1, it lead to the mutation of the genetic material. Don't forget, my dear student, this ratio will be attained in where, low in microorganism and high in the higher animals. 0.4 considered as a low microorganism. Take an example, the bacteria, E. coli, lambda, bacteriophage, whatever it may be, fungi sometimes. And higher animals, 1.9, it may be higher, means take an example, chimpanzee and other animals also, dinosaur, whatever it may be vary. Okay, this is also a important thing. See, this is the scientist of Watson Crick. Name of the scientist, Watson Crick, is an old person, but he discovered the double standard helical structures and also constructed by which one, nitrogen bases, sugar molecule, and also phosphate. And the nucleotide is also there, number of nucleotide. Okay, he stated the statement and structures also. It described the DNA molecules. Okay, see that one. Detailed structure of the DNA. The DNA molecule contain what are the part is that. Already we discussed simply, but here we are going to discuss very deeply. Okay, clearly watch it, my dear students. DNA polynucleotide chain has two free end. Already told you. One is 5' end and another one is called as 3' end. The free phosphate moiety at 5' end. Note it down, my dear. This is very, very important. 5' end always end with the phosphate moiety, otherwise called as phosphate edge of ribose sugars. Okay, 5' end. The 3' end always end with the which one, ribose sugar. Don't forget, my dear. 5' end phosphate. 3' end which one, sugar molecule. So DNA, it contains 5' end, 3' end. 5' end always end with the phosphate moiety. 3' end always end with the ribose sugar. And free 3 OH group of ribose sugars. This is also important part. Watson and Crick were the first to propose the double helical structure of the DNA based on the X-ray diffraction technique. Note it down, my dear student. How the DNA molecules was, what is that? Proposed by the Watson and Crick. This is also another question, 2019 or 20, I think. They asked the questions. Which based on the X-ray diffraction technique. Which technique they were used by Watson Crick, means the X-ray diffraction techniques. This was given by the name of the scientists. What is that? Franklin and Wilson. On the basis of Franklin, X-ray diffraction was discovered by the Franklin and Wilson. Without these scientists, what happened? Watson, they will not discover these structures, DNA structures. Yes, Ajay? Any doubt?
Sir,
Yeah.
sir, could you explain this X-ray diffraction technique, sir?
It's X-ray diffraction means only the X-ray methodology. What is that? What the structure is present? I will tell you, but at the same time, X-ray means what is the inside? What are the component present in it? Exhibit. X-ray
Yes, sir.
diffraction means separation. Diffraction means the nitrogen bases, how they will be split, how they arranged. That things can easily identified with the help of this technique only. How the bone are broken, how will you identify? With the help of the X-ray. They were used the X-ray, rays used for the identification of the nitrogen molecules. How the base pairing is arranged. This part. What is that? This part. See that? This part. How it will be arranged like this. This part identified with the help of the X-ray. This is the structure. In between, how it will be present in it. They analyzed, they observed with the help of the X-ray diffraction method. This was done by one of the scientists, this structure inside. I will tell you remaining things. What is that? X-ray diffractions in next classes. Okay. This is the basic method. They used the X-ray rays for the identification of the internal structure of the DNA molecules. Okay. That is X-ray diffraction, I think. So otherwise, I will tell you a clear idea next classes. Okay. See that?
Yes, sir.
Yeah. Two chain complementary to each other. So already in our report. Two chain is there, already discussed. There is a two chain is there. One is started with the 5 dash n, another one is a 3 dash n. This is this chain is also combined with the another complement, is a near one with the help of bond, already we discussed, isn't it? That is called as complementary. Another started with the 3 dash n to 5 dash n. Okay. See here, adenine thymine pair has 2 hydrogen bonds. See that? 2 hydrogen bond is there. Adenine molecule, this one, and this one is thymine. So there is a bond between these two. That is a hydrogen bond. So this is also one of the questions. I will tell you how it will becomes. Another one, guanine and cytosine. The pair has a 3 hydrogen bonds. See that? 1, 2, 3 hydrogen bond. Guanine and cytosines. Okay. This is the occupied in the center of the which one, you know, nucleic acid, DNA molecules. Here addition with the sugar molecule and also phosphate. Here addition with the sugar molecule and phosphate. This one base pair connected with the another one. So likewise the polynucleotide chain formation takes place. Okay, see that. Upon heating at temperature above 80 degree to 90 degree, note it down, my dear students. Upon heating, okay, 80 degrees Celsius to 90 degrees Celsius, what happened? The two strand uncoil and separate. The condition is called as denaturation. As a result, you will get 2 single strand DNA molecule. Already told you, isn't it? On cooling, so here the spelling mistake is the clouding. The condition is also clouding, otherwise called as cooling process. 2 strand joined together. Okay, there is comes out of the heading, I think it's minus nature. Okay, this is the 90 to 80 degrees Celsius. When you are going to heat the DNA molecule, what happen? It will be denatured. That bond will be broken. As a result, they will be separated or uncoiled in 2 single strand. Suppose we are cooling, what happen? You know, it will be rejoined together. Okay, this is very, very important. And another one is also very, very important one. DNA, it exists in the 4 types of. One is called as DNA A form, DNA B form, DNA C form, DNA Z form is also there. See here, DNA mostly right-handed and B form. Don't forget, my dear. This is very, very important. This is also 3 times they were asked in the NEET question paper. DNA mostly right-handed and B form structure. Okay. Z is also having how they will be classified A, B, C. You know, the presence of the which one, you know, nitrogen bases and the rotation of the right side or left side rotations. So this is DNA mostly right-handed and B form only. The Watson and Crick also discovered the which form, you know, right hand B form structure. The bacterial nucleotide consists of a single circular DNA molecules. What is that? It consists of circular DNA, cDNA. Sometimes they may give a small c and DNA like that, you know, is C denoted as circular DNA. Okay, don't forget, my dear. In this part, you must note it down. 3 points is very, very important. One is called as 90 degree, up to 90 degree denaturation occur. It will give 2 strands. Cooling condition, that condition is called as annealing. So it will be united together or rejoined together. Okay, and also get original positions. And the DNA mostly in the right-handed and B form structure. Note it down, my dear. And bacterial nucleotide also consists of single circular DNA molecules. Bacterial nucleotide. Okay, this is also very, very important. See that DNA double helical structures. DNA is composed of 2 polynucleotide chains and large macromolecules. It is twisted anti-parallel chain held together by hydrogen bond. Sugar, phosphate forms and the backbone. Sugar and phosphate forms the backbone of the nucleotide. Nitrogen bases forms the interior pad through hydrogen bond. The covalent bases bearing is an important features of the DNA structures. These are the combination. Particularly, the nitrogen base is united together 2 strand. Already you know about nitrogen strand is very, very important. See here. Already we discussed, See that, this one. This is the sugar molecule. This is another sugar molecule. In between, this is a nitrogen bond. Without nitrogen bond, what happened? The two stand, it will not unite. Okay. The two stand, it will not join together. How it will be joined? Because the presence of nitrogen base with there is a bond in between 3 nitrogen, 2 nitrogen bond. That gets expressed in the point. See this one. See this one. Okay. Covalent base and also the bearing important feature of the DNA molecules. This is the nitrogen base forms the interior designs paired through hydrogen bond. Okay. Go to the next one. The two polynucleotide chain have an anti-parallel polarity. Anti-parallel means 5' to 3' end. 3' to 5' end. Okay. The two chains are called in the right-handed fashion, forming a right-handed helix. Right-handed means about twisted like this. Right-handed side. Suppose if it is left-handed means levorotatory and dextrorotatory like that. Okay. Uniform distance is maintained, very, very important between the two stand-off helicals. Uniform distance. It will not the improper manner. The correct formation, it will be present, and the uniform distance is maintained between the two stand of the helical structure. How it will be, these are the point very, very, very important. Each and every tan has a 10 base pair. Don't forget, my dear. Each tan, see how many base pair is that? 10 base pair is there. See here. So, okay. See, just I draw a small diagram here. So consider here. Okay. So this is the one tan. This is the starting point. This is the one tan. This is the another tan. This is the first tan. This is the second tan. This one is a third tan. Each tan in between, what is that? 10 base pair. 10 base pair is there. 10 base pair. Okay. 10 base pair means what is that? Adenine. Same things. Thymine. And guanine with the cytosine. So likewise, what happened? It arranged. So in this part only 10 base pair is there. That is called as 34 Armstrong. 34 Armstrong. So total the 10 base pairs in between. This rotation is how many base pair is there? In this part 10. So in this part is also 10. In this part also 10. So that is a equal proportion is there. Each rotation there is a 10 base pair is there. It consists of 34 Armstrong. Okay. See here, another one is very, very important. So adenine molecules is the, imagine, combine with the thymine. Okay. So guanine molecules combine with the cytosine. So in this part, in between these two stand, there is a space is there. The two molecules, 1 to 10. Okay. From this is considered as a first one. This is considered as a second one. Likewise, how many molecules there? 10 molecules are there in this part only. Okay. The adenine, thymine, guanine, cytosine. In between these two nitrogen bases gap is there, 3.4 Armstrong. 3.4 Armstrong in this part. Okay. Adenine and guanine. There is a space is there 3.4. So how many molecules present in it? Each rotation there is a 10 is there. Isn't it? Each rotation there is a 10. In between this gap is now 3.4 Armstrong only. So you multiply 3.4 into 10, you will get 34 Armstrong. So this is very, very important things. This is very, very important. Don't forget, my dear. So this is very, very important. So once again, okay, I will show you. Are you clear in this area, my dear? Ajay?
Yes, sir. Yes, sir. Clear, sir.
Okay. So each and every rotation, how many base pair is there, my dear? Each base pair. Each rotation contain how many base pair is there? Yeah. Tell me, my dear. Each base pair, each rotation. See that once again. Okay. This is called as a one rotation. This is a one rotation. This is okay. In between.
Sir, can you hear me, sir?
Yeah.
Sir, yes, sir. Sir,
Ah.
10 base pairs.
Very good, my dear. 10 base pair is there. Each nitrogen base, there is a space is there. That space is how much, my dear?
Sir, 3.4 angstrom.
Very good. Exactly correct. 3.4. See, 10 base pair contain 34 Armstrong. Each nitrogen bases, take an example, first nitrogen bases as well as the second one and third one and fourth one up to 10, isn't it? So in between there is a space is there. That space is how much, 3.4 Armstrong. The 3.4 into 10 nitrogen bases, you will get, what is that? 34 Armstrong. Okay. So don't forget, my dear, this is very, very important. Okay. So we'll pass on to the another one. So I will finish it up to this level. I think it's better for you. Okay. Sorry. Okay. Okay. This one I will finish it when. Okay, my dear. So, okay, this one. So each tan has a 10 base pair, 34 Armstrong. In between two nitrogen bases, 3.4. Okay. Diameter of the helix, don't forget, my dear student. Diameter means in between, isn't it? In between the one phosphate molecule, sir, and another phosphate molecule. One sugar molecule to another sugar molecule, there is a 20 Armstrong is there. Don't forget, this is also very, very important. DNA found both nucleus and cytoplasm. And extra nuclear found in the mitochondrial and also chloroplast. This is also very, very important one. Okay. So next one is, what is the packaging of DNA molecules? We will discuss in the next classes, my dear students, because up to this level is very, very important. Particularly DNA structure is very, very important. Okay. So what are the things already we discussed? What is meant by nitrogen bases, how it will be formed, and also the Chargaff rules is also very, very important one. See that one? Chargaff rules, who stated the statement, Chargaff. That's why they have given us as Chargaff rules. One adenine molecules always combine with them another pyrimidine. Adenine molecules always equal to the another pyrimidine molecules. One adenine, see that one, see? Adenine, thymine. So these two are equal. Adenine is a, what is that? Purine molecule. Thymine is a pyrimidine molecule. So one adenine combined with the another pyrimidine molecules only. So these two are equal. Number of molecules, adenine, how much is present in it? It is equal to the thymine. Guanine is also equal to the cytosine. This is the rule of the, what is the Chargaff rules. Okay. Okay. I will summarize and within 2 minutes. What is the things already we discussed? So what are the bond is present in it already, but there are 3 bonds have been reported here. So the formation of polynucleotide, how it will be? N-glycosidic bond, phosphodiester bond. Okay. So how it will be? Already we discussed the slides, isn't it? So go to the next one is what is already we know what is the difference between the DNA and RNA according to the nitrogen bases and also structures. DNA molecules is the double stranded structures and RNA is a single stranded nucleotide. But here there is a adenine, thymine, guanine, cytosine also present in it. But instead of thymine, the uracil only present in the RNA molecules. Okay. That things also already we discussed. How it will be bonded with the nitrogen bases. Guanine scenes combined with the molecule cytosine. triple hydrogen bond. This is also max oriented calculation. I will tell you how it will be in future. Okay. Adenine combined with the molecules of thymine with the 2 hydrogen bond. So these are the bonds. When you are heating up to the 90 degrees Celsius, this bond will be completely broken. So the 2 molecules will be separated. Okay. Another one is also a phosphodiester bond. One sugar molecule, also another sugar molecule bonded with the phosphate molecules. That's why it is called as a phosphodiester bond. The bond is here one also one ester bond. Here also one ester bonds. 2 ester bond with 1 phosphate molecules. That's why we connected the 2 sugar molecules. That's why they have given as a name is called as what is a phosphodiester bond. Okay. Okay. See, these are the nucleotide and nucleoside formations. Already we discussed how it will be. Okay. And then, so this is the entire structure of the molecules. See, in between there is a 10 molecule is there. Already we discussed each and every nitrogen base. There is a space is there. That space is called as 3.4. The diameter of that, this one is diameter. This diameter is 20 Armstrong. This 2 nitrogen base, there is a 3.4 Armstrong. Each rotation, it has 10 nitrogen bases, total 34 Armstrong unit. Okay. So anti-parallel each other, 5' and 3' 3' and 5' like that. Okay. So Chargaff rules already we discussed. Okay. Suppose if it is any variation is there, lower animal as well as the higher animals, lower plant or higher plants. Okay. It may be vary. Okay. Due to the, this is the scientist name, Watson and Crick. Okay. The father of genetics. Okay. Father of genetics. Who is the father of genetics? Father of DNA molecule. Okay. Double standard DNA molecules. Detailed structure also we discussed, particularly X-ray diffraction techniques. It's very, very important. This was introduced by the name of the scientist. What is that? Wilson and Franklin. Don't forget, my dear students. And Griffith rules. So I mean, Chargaff rules also very, very important. Chargaff rules nothing but adenine, thymine, 2 bond. Guanosine, cytosine, 3 bond. Okay. 1 purine, another pyrimidine. Okay. Number of molecules adenine equal to thymine. So these are the rules of what is that? Chargaff rules. Okay. Thank you so much for the participation. We will discuss the remaining things on the next class, my dear students. Thank you. Bye. Take care.
Thank you, sir.
Thank you, Gunna. Thank you, ma'am.
Thank you, sir.
Thank you, ma'am. Okay.